Hello, everyone. Today I'm going to talk about my RAP2 project, Characterization of Ventricular Volume in Junk-Deficient Brains. First off, let's ask, what is junk? Junk stands for C-Jun N-terminal kinase. It is a critical signaling pathway that regulates neuronal function. The junk pathway is regulated via 3 junk genes, named junk 1, 2, and 3 respectively. Loss of this function has shown to lead to disorders, such as autism spectrum disorder. To better study junk, our lab used conditional genetic deletions to remove junk function from the ventral forebrain to study its effects on the developing brain. In this model, junk 2 and 3 were knocked out of the whole mouse, while junk 1 was only knocked out of the DLX 5-6 territory, as shown in green, meaning only the green territory is completely knocked out of all 3 junk genes. From previous work done in the lab, it has been shown that mice with junk triple knockout show enlarged ventricles. In this study, we wanted to develop a quantitative way to measure these ventricular volumes to better understand the relationship of the control brains and our triple knockout. Next, we can see the total methodology for how we turn these mice into quantitative data. We started off by mating the mice that will either express a control genotype or a triple knockout genotype. The pups were then collected at postnatal day 0, which is the day of birth. They then had to be preserved using a fixation, and the mice were embedded before the brain was placed into the scanner. Afterwards, the heads were scanned using a micro CT scanner, which allowed us to view, in high definition, the entirety of the brain, without damaging the brain. By referencing brain structures with the Allen Brain Atlas, shown in picture C, the ventricles were traced by hand in order to form isolayers using the program called Seg3D, which in addition was able to calculate ventricular volume. These are the results we found. It was determined that not all triple knockouts had the same effect. Some were more severe than others. I've shown some models that express mild effects on forebrain development and some that express severe effects. Each brain has a segmented view showing what its ventricles look like in the sagittal, coronal, and axial position. The fourth set of images, however, is the rendered model of the ventricles made by the Seg3D program. By viewing the images, it can easily be concluded that even mild triple knockout has more ventricular volume than the control. But the question is, by how much? To answer this question, we must take each ventricle into account and subcategorize. From the left to the right, we can see 3D rendered models of the lateral, third, and fourth ventricles. The number of brains we had for each genotype was 3, meaning we had 3 control brains and 3 triple knockout brains. As you can see, the triple knockout lateral ventricles have much larger volume. Although the p-value is high on the triple knockout, even the lowest point is still much higher than the control brain. The third ventricle's volume differed the most from the control counterpart. It was shown to have the greatest increase in volume between genotypes. Finally, the fourth ventricle showed the least change between genotypes, although it did continue to go with a trend, and it showed higher volume in the conditional triple knockout. It should be noted that none of this data was shown to be statistically significant. However, this is more than likely due to the small sample that was used in the experiment. In future experiments, we'll have greater sample size. It was overall shown that the conditional triple knockout for the junk genes create enlarged ventricles within our mice model. And then in the future, We will use this to analyze techniques to measure specific parts of the mice brain in order to better determine how the function of junk affects cortical and subcortical neuronal pathways. Thank you to the RAP program.